Hi, welcome to this video on enzymes. Now, the first thing is, what are enzymes? Well, enzymes are biological catalysts. They are biological catalysts, and they catalyze every metabolic reaction in the body. They're also made up of proteins. They are proteins. Sometimes they have non-protein structures, but all catalysts, all enzymes are made up of protein molecules, and they act on substrates. So the molecules that they break down are called substrates. So they act on the substrates, and they have active sites. That's the site on the protein molecule of the enzyme that does the splitting up of the substrate, and these active sites are three-dimensional, and that's what you need to know about enzymes in terms of what they are. And this lesson is going to go into some more detail about the specificity of enzymes and also how they are inhibited. So specificity means that enzymes only work with specific substrates, and what we're going to have a look at is the lock and key model, which shows how specific enzymes break down biological molecules. So here we've got a schematic of enzymes and substrates. So let's first start by labeling it. This one down bottom left is a schematic of an enzyme, and this sort of region here is the active site. We try to depict it here in 3D, and this would, on the top left hand side, would be the substrate. So that would be the substrate, and in this case we're looking at a sucrose molecule. And you may know that sucrose consists of a glucose and fructose molecule bonded together, where this would be the bond. So as the red arrow shows, the sucrose molecule and the enzyme bind together, and when they are bound together, they form the enzyme substrate complex, which is this one in the middle. So let's label this one in the middle, the enzyme substrate complex. When the substrate and the enzyme bond together, and then what happens is that the binding of the substrate and the enzyme, it places stress on the glucose-fructose bond. That's this bond here. That bond is stressed and it breaks. You can see no bond here. So let's write that down. So the binding of the substrate and enzyme breaks the bond, breaks the glucose-fructose bond. Okay, so then that leaves the products, glucose and fructose. So let's say this one is glucose, and let's say this one is fructose. That leaves the products free to go. They are released. So products are released. And as you can see, the enzyme is unchanged. So enzyme unchanged, just like catalysts are all unchanged. So the enzyme is free to go off and bind to other substrates. And that's how the model works. Hopefully that's been clear for you. But one other point that's worth making is when we're talking about specific substrates, that even applies to enantiomers. So that means that they're stereo-specific. Stereo-specific. So they only work on one enantiomer. So if the other enantiomer comes along and tries to bind into the enzyme, then it won't work, they won't fit. Just sort of like how if you try and put your right foot into a left shoe, it's not going to fit. So you need your enzyme to be even specific to that extent. Okay, at the start of the video we mentioned that we were also going to talk about enzyme inhibitors, and these are molecules that have a similar shape. So they are molecules that have a similar shape to substrate molecules. The molecules that have a similar shape to substrate molecules. And if they've got a similar shape, they can react or interact at least with the active site of the enzyme and stop the enzyme working. And that's useful in certain circumstances. For example, drug manufacture, which we'll have a look at in a minute. But let's have a look at this process of inhibition. So if we're here, let's have a little picture of an enzyme. This is our enzyme, and that's our active site. And so let's have a look at a substrate molecule. This green one is the substrate molecule. And like we said above, that the molecule that is the inhibitor has a similar shape. It certainly has a similar shape as far as the bit of the molecule that goes into the active site is concerned. So this red one, let's call that the inhibitor. And what happens is, if you've got enough of a concentration of the inhibitor, it would join with the active site of the enzyme, like so, and therefore you would block the substrate from getting in there. That route would be blocked, and so that reaction couldn't happen. And the amount of inhibition that occurs is dependent on the concentration of the inhibitor versus the concentration of the substrate, and also how strongly the inhibitor bonds to the active site of the enzyme. If it bonds very strongly, then it's difficult to remove, and that would inhibit more reaction than if it bonded weakly. And I just mentioned that sometimes this is useful. It's useful in drug manufacture. For example, the drug Tamiflu, which is an anti-flu drug, works in this way, where the drug in Tamiflu acts as an inhibitor for an enzyme in the virus that helps the virus to create its cell walls. And so without being able to create the cell walls, the virus can't replicate, and therefore your flu is aided. Now this process of finding a molecule that fits into an active site of an enzyme is very complicated and takes a long time. It's mostly done by trial and error by the pharmaceutical companies, and so they have to try hundreds of thousands of molecules to find one that fits nicely enough into the active site of an enzyme that they're looking for. They can test different molecules very quickly. You'll have seen those multi-test tube machines that are automatically filled up with different chemicals, so they can test it quite quickly, and they can also use computers to model the active site of the enzyme to sort of narrow down the shape of a molecule that might fit into it. So they narrow it down with computer modeling, and then they're able to test potential molecules in large numbers quite quickly. But still, nevertheless, it takes a long time and takes a lot of money. So that's why pharmaceuticals per gram are so expensive. And it's especially difficult if the drug molecule is chiral. Then only one enantiomer will fit into the active site, because the active sites of the enzymes are usually stereospecific, as we said.